You know that Dr. Sebastian Haidt was supposed to be the last speaker, but unfortunately his plane got delayed, or actually canceled, and he is now flying somewhere up there. So we were very fortunate to have Dr. Ahmed Mustafa, who's going to fill in for him today. So Dr. Mustafa is an associate professor at the University of Saskatchewan College of Medicine and Provincial Health Care and co-lead of genomics of the Saskatchewan Health Authority. With over 5 years of experience directing the histocompatibility and immunogenetics lab at St. Paul's Hospital, his work focuses on clinical research and molecular biology to improve patient outcomes. Dr. Mustafa is certified by the American College of Histocompatibility and Immunogenetics and is committed to empowering healthcare systems with innovative solutions to meet complex medical challenges. So please let us welcome Dr. Mustafa.
Thank you, Dr. Murphy, for this nice introduction. And I'm actually very sorry to disappoint you. I'm not Sebastian. So just the green button, I guess. No, I don't think this is my slides. Can someone double check the slides again? Yeah, that's not my slides. Yes, that's my slides. Okay, so this is my actually disclaimer slide. And as you know, I'm only here to save the day. This picture was shared to us by Dr. Sebastian yesterday. His flight did a U-turn in the air 2 hours after departure, and they have to land back again to his home, and he are able to do it today. So hopefully he will arrive at 9:30. But it's too late. I did a little bit of modification from his slides, so just like to be my style a little bit. So I was actually looking to see why did they select me to give this talk, and since we are actually talking about molecular matching, I started to download the image for Sebastian. And then I found that we're actually matching in some things and we are mismatching in other things. So I think that was the reason. So I only received this late notice at 10:30 p.m. yesterday. And Dr. Oscar told me, yes, you can do it. And then he said me, at the beginning, I thought I'm going to present my own work. But then he said, no, you're actually going to present Sebastian's work. And then he sent me the slides. And then I looked at the slides 1 hour later. I called him back again. I said, I couldn't do it. It's so confusing. So I remember another phrase from Dr. Oscar on a completely different occasion. And he told me, if you cannot convince them, confuse them. So if I am able to confuse you today, then I'm successful. So this is actually the donor kidney cell, and the donor kidney cell will express eventually a mismatched HLA antigen. So the patient will see this mismatched antigen as a foreign, so the antigen-presenting cells, in this case is the B cells, will recognize this mismatched antigen, and it will start to bind to the B cell receptor, and then this antigen will be internalized into this antigen-presenting cells, get chopped into small pieces, and then it will be presented in the context of HLA class 2. This will eventually activate the CD4 positive T cells, which in turn will activate the plasma cells to produce antibodies, and this will result in antibody-mediated rejection, or it can actually activate the CD8 positive T cells, and this will result into cell-mediated rejection. And for the purpose of today's presentation, I'm just going to focus on the B cells and the T cells because they recognize different epitopes on the same molecule. So here, if you have a patient typing on the top, and these are all possible donors, the patient will see this donor as a mismatched antigen. And the reason, because our immune system is programmed to recognize allorecognition because it is based on amino acid disparities. So now we have multiple software algorithms that can actually measure and predict this amino acid disparities. And this is just like a snapshot of the software that we have so far. I might be missing one or two. But the first and the old one, and already Dr. Oscar has already mentioned it, is the HLA matchmaker, which actually look at the different eplets between donor and recipient. And now this HLA matchmaker has not been updated for a very long time, so it has been mostly replaced by the HLA Eplet registry because it is more updated every quarterly. And then there is also the EPVIX, which all these software look at Eplet mismatch. Another category which has been developed by the University of Cambridge, and they actually look at the electrostatic mismatch. So whether it is looking at the amino acid or looking at the HLA molecule itself, they're still the same scenario. And then later on, there is another software that actually look at single amino acid on the extracellular domain. And I'm going to give you a little bit of details of these three different programs. And finally, we have our PIRCHE, which up to this date, at least at my knowledge, this is the only software that look at the T-cell mismatch or the T-cell epitopes. So if we are looking at the Cambridge HLA AMS, so it actually compared the donor and the recipient HLA extracellular domain amino acid sequence, and then count how many amino acids differ between them. So for example, the figure you see here, that if you compare this donor to these two patients, you can see that the donor has two mismatched amino acids. So now this gets a score of 2. The other software developed by Lydon is the HLA-EMA. And this software actually compares solvent accessible amino acid mismatch and then total amino acid mismatch between mismatched donor HLA alleles and the corresponding recipient HLA alleles. So basically, it's a software. In the top, you put the patient typing, and in the bottom, you put the donor typing, and then hit the submit button, and then this table will show up. It will show you the total amino acid mismatch, and also it will give you the solvent, because not every amino acid is actually accessible. So it has to be accessible so that the antibody can see it. And the PIRCHE-B, which was known with SNOW, but this is now a complete rebranding of the name. So they changed the name from SNOW into PIRCHE-B. This software is actually unique on the way it actually looks on data. So it followed two different algorithms. The first algorithm is called the snowflake, and then the second algorithm is called the snowball. And I hope, Matthias, I can see him here. I hope that I am presenting it right. So it actually calculates the solvent accessible surface area for every residue, and then it selects the residue above a certain exposure threshold. So this is the first phase of it. And then the second phase, it actually looks at the geometry or the shape of the antigen, because antibodies does not like flat surface. Antibodies actually like convex, concave surface, not even convex. So it filters this as accessible into another layer, and then it gives you the score. And now we have these three different software. All of these softwares are actually clinically relevant, because all of this software has published data to show that they are actually working. So if we look at the AMS score here, and then if we focus on the bottom line here, you can see that these are the patients who are very, very high risk, and they have the highest AMS score. So it looks like the score is working. The other thing here that you see is the HLA-EMA. And this was a study looking at the scoring of the HLA-EMA, and they found that the more score that you have, the more prediction that you are going to develop antibodies against the DU. And then finally here you can see the SNO or the PIRCHE have highest score as a predictor of donor-specific antibody. So the important thing about this idea is you need to link this B cell epitopes to what we call the T cell epitopes. So for example here, you have the donor kidney cell, and this is the donor kidney cell expressed, I would say this is a class 1 because I think I missed the beta-2 microglobulin when I was drawing it, but trust me, this is a class 1. So at one point it will get shed out, and this will be presented to the patient antigen-presenting cells through the B cell receptor group, because the B cell receptor, which you see in green here, is the surface epitopes. And then it will be internalized, and then now you will see that this core epitope, which was present in the core not accessible, now become accessible, and then this will bind into the context of the HLA class 2, and then this will activate the CD4 positive T cells. And the idea of this PIRCHE is not just like, it's an older idea, and it actually started with this publication in Human Immunology in 2004, where they were looking at HLA-DR phenotype is a predictive of humoral response against the class 1 antigen. So basically what they did is they took two cohorts, a cohort that express or have frequently produced HLA antibodies, and another cohort that produce less frequency antibodies. And then they looked at specific typing of the patient, and then they looked at the typing of the donor. So for example here, which I'm highlighted in red, you can see in the cohort that have the antibody produced more frequently, if you look at the DR4, you can see that if the donor is B7, there is high likely that there will be a development of a donor-specific antibodies against the B7. And if you look at the other cohort, if the patient is DR4 if the patient is DR4 and the patient is B35, they are unlikely going to form a donor-specific antibody. Because here we need the DR4 so that it can present the peptides from the class 1 to the CD4 positive cells, so to activate the plasma cell to produce antibodies against them. So here you can see that the one on the left side is actually a deleterious antibody, and the one on the right side is actually a protective one. And PIRCHE, which also got rebranded now, so when you read newer papers, it will be called PIRCHE T2. So it can differentiate between the PIRCHE B and the PIRCHE. So the PIRCHE, or the PIRCHE 2, is basically, this is a mismatch donor HLA, and then it entered the antigen processing pathway. It got chopped into multiple peptides, and then this peptide will be presented in the context of HLA class 2 from the patient. And then because of the idea of MHC restriction, not every single peptide that are digested will be presented. So for example, here you have 5 different peptides, but only 3 of them is going to be presented. And then this will get a score of 3 if you're using the PIRCHE. And then this is a newer software which is unpublished yet. It's called NET MHC PAN, and it works in a very similar scenario as the PIRCHE T2, and we will see in the next slide. So here I will show you that there are model performance for single HLA immunogenicity, and I'm going to take the HLA-DQ as an example here. So here I just want to show you that even every software have a good predictive value, because here you can see the area under the curve is almost close to 1 if you are using the AMS. And also if you are using the other algorithm, it is still the same. And this is the newer model for T-cell receptor called the NET MHC, also have a very good prediction of rejection. But what I want to stress is that it is very important to use the threshold, because here we are using 1,000 nanomole threshold, and then this is what you see with the 1,000 nanomoles. This means it's a very low, very high, anything below this threshold will be presented. So it will present a lot of these T-cell epitopes. But if you use a 500 nanomole threshold or even a 50 nanomole threshold, this prediction will significantly decrease. So be very cautious when you are using all these types of algorithms, because you need to understand the biology behind it before looking at the numbers. And you can see here that the PIRCHE also used the 1,000 nanomoles. So the PIRCHE is actually, at least this is my understanding, and maybe Matthias can correct me. And looking at, I think, correlation between all these software, and my colleagues already presented, so I'm going through these slides so quickly. But here you can see that comparing the amino acid to the Eplet, there was very strong correlation. And this study was published by Webel in Transplantation 2018. And then here another study looking at PIRCHE, and again as the different mismatches, and again the very strong association. And a third study showing the EMA with the PIRCHE, very strong correlation. And using the EM3SD score against the amino acid mismatch, strong. And finally here the EMA against the mismatch. So I'm not trying to tell you that one software is superior than the other. You can see there is tons of publications showing that they are all acting in a very similar way. So there is another group who actually published that combining the scores as well. So here they combined the DR with the DQ, and here also they combined the total score. So the problem with this, when you do combination, yes, you look as improved predictive value, but it's actually the specificity and the positive predictive value remains very low. So this is very important to be cautious, because our immune system does not see numbers. Our immune system sees biology. So if you have a high score or a low score, our immune system doesn't understand that. So for example here, if you look at this cohort, so the one on the upper right quadrant, this means that both the PIRCHE and the EMA are the high score. So you can see most of the donor-specific antibodies are in this quadrant. But look at these two guys here. The score is still low, but they still develop donor-specific antibody. And that's why we need to understand how this works, because if you look at the B cell and the T cell, they are not an additive effect. They are actually work together. They are linked together. And this is why it is important to understand the biology before applying these scores. And here, if you have a patient, and this patient have multiple donors to pick from, and you can see, that the first one is the easy one, because the first one is the exact match. But now you have three different donors to pick one. Which one is better than which one? If you're using just like the Eplet mismatch, it will tell you that all of them have one mismatch, so they all look similar. But now you need to use multiple or combination of other approach. So for example, if we look at the physicochemical disparity, you can see the first two get no and the second two get yes. If you look at the T cell helper, the first and the third will get no, and the second and the fourth will get yes. And then if you look at the prediction of immunogenicity, you will see that the only one that is highly immunogenic is the last one. So here this is how, if you want to use multiple algorithms, you need to understand each one, apply each one individually. And this is a nice study published in the 18th International HLA and Workshop Immunogenetics. And they were looking at, because not all eplets are created equally. Some of them are really immunogenic, some of them are less immunogenic. So here they found that like the top ones here these are the ones that are really immunogenic, and the bottom one are the ones that are less immunogenic. But what actually I found is very interesting, and I don't know if you actually notice it, is the immunogenic one are the ones that are actually less expressed than the low immunogenic. So I would think that if the epitope is more expressed, this means it's more immunogenic. But here, it changed my idea about how immunogenicity is counted. So, and also, it's not only restricted to B cell epitope, that it's also on the T cells, they are not equally immunogenic. And this is a big, huge study, used 118,000 patients, and they found 1,935 different PIRCHE, but only 86 of them were immunogenic. And they were associated with sensor graft failure. So in conclusion, mismatch correlate with the DSA formation on a population level. Different mismatch have different similar performance, while combining mismatch algorithm may increase the predictive power. True linked recognition has not been studied yet. And if you, in this area, and you are looking to do some research, take these ideas and start your own research. Immunogenicity of specific amino acid mismatch, the effect of non-exposed amino acid residue on immunogenicity, the B cell receptor and the T cell receptor variability and interaction with allogenic HLA, the actual allopeptide generated in the endosome compartment for HLA class II loading, and the role of affinity of HLA-derived peptide to HLA class II, and finally, amino acid disparities truly generate a T cell response. I think I will stop here, and I think I'm done.